The neuron is constructed of a cell body and two extensions, the dendrites and the axons. The dendrites function as a sensory organ, receiving electric signals and directing them to the cell body, and the axons transmit the impulses from the cell body outward to other neurons' dendrites. Many axons are wrapped in a fatty sheet called the myelin sheath, which speeds up electrical impulses. The gaps between the myelin sheath are called the nodes of Ranvier. Saltatory conduction is the process of electrical signal jumping from node to node, causing the signal to accelerate. The axon splits into terminal branches which branch off for more contact with other dendrites.
There exists a membrane potential, which is the difference in electrical charge between the cytoplasm of the cell and the extracellular fluid. The resting potential of the membrane is formed by the sodium-potassium pump. It moves sodium ions to the outside of the cell while potassium ions are brought into the cell. This causes the inside of the neuron to be very different from the outside. There are more potassium channels open at this stage than sodium channels. Because potassium begins to diffuse at a greater rate and the cell is not permeable to chlorine, the inside of the cell has a negative charge while the outside of the cell has a positive charge. In order for the nerve to fire, the stimulus must overcome the threshold. This overcoming would be called depolarization, and the inside of the cell is made less negative relative to the outside than at the resting membrane potential because voltage-gated sodium channels open. This triggers a positive feedback loop that causes more sodium channels to open, causing the inside of the cell to become less and less negative.
After reaching the action potential, the voltage-gated sodium channels begin to close while the voltage-gated potassium channels begin to open. This causes the interior of the cell to have a more negative charge while the extracellular fluid has a more positive charge. This would be known as repolarization, change in membrane potential that returns it to a negative value. Following the repolarization of the cell, it enters the undershoot, or hyperpolarization, where due to the sustained opening of the voltage-gated potassium channels while the voltage-gated sodium channels remain closed, the membrane potential has a greater negative charge than at the resting potential. During this period, known as the refractory period, another action potential cannot be reached because the stimulus cannot reach the threshold. This prevents signals from traveling backward on an axon and makes them only go in one direction.
After traveling electrically along the axon, the synapse is crossed chemically by the impulse. The synaptic vesicles are located in the cytoplasm of the presynaptic neuron, the neuron before the synapse, and these vesicles store neurotransmitters that cause transfers of the impulse. These neurotransmitters can be inhibitory, causing a negative change in the membrane of the postsynaptic neuron that it joins to, or excitatory, causing a positive change in the membrane of the postsynaptic neuron that it joins to. When depolarization of the presynaptic membrane occurs, Ca2+ ion levels increase, causing the vesicles to merge with the presynaptic membrane. The neurotransmitters are released into the synaptic cleft, the space between the neurons at a nerve synapse, where the nerve impulse is transmitted. The neurotransmitter attaches to receptors on the postsynaptic membrane, causing a response.